Okay, so now see, this is, I told you about the fluorescent staining is commonly used to improve tuberculosis diagnosis. Oh, this is not. Okay, so this. So principle of fluorescence, it is that when light radiation of high energy strikes a substance that can fluoresce, the substance absorbs that energy and converts a small part into energy, that is heat. So light higher energy strike on the some fluorescent molecule, so it will absorb energy and convert a small energy. And the energy is not absorbed by the substance, is emitted. The small part of energy which couldn't absorb by the substance again is emitted again as a light, and this emitted light it is called as a fluorescent light. So here it is a fluorescent light. So you use some light source and you give some energy. So after this it will absorb certain light, but it will start emitting certain energy loss, and this you will see in the form of another color. So this is emitted as a this fluorescent light, and it's generally it is this 400 to 700 visible range is very important in case of fluorescent light, because you can see it clearly by your naked eyes. Now there are two different type of fluorescence. Substance that can be activated to fluoresce all called fluorochromes. So fluorochromes means they give color by using fluorescence phenomena. Fluorochromes may be naturally present in biological material. I told you that it can be present through the biological material. So these are the primary, or might be artificially introduced into these materials. This is known as a secondary fluorescence. It's important, but it's okay. I already explained you about the basic fluorescent microscope part last time, that there is an emission filter, there is an excitation filter. The important thing about the emission filter and excitation filter is that it will pass on particular wavelength only. So that's why these filters are put into the fluorescent microscope. And dichromic mirror, I already explained all these things. But if the next thing is fluorescent microscope. So generally these days this is fluorescent microscope is two different types. One is a transmitted light, one is a EP illumination. These days this EP illumination is much used in research. So now what is EP illumination, or what is in a transmitted light? So it is again very simple. The excitation illumination through the specimen. Okay, what does it mean? Usually from the below. So from below you are putting the excitation light and it is passing on the specimen. I will show you. And EP illumination, incident light shining on the surface of the specimen. Okay, so let's understand this one. The transmitted, here you saw this is the light source and this is the excitation filter. So what excitation filter will allow? It will allow that excitation light will only go. Okay, and this is your sample. Where is your sample? Yeah, this is your sample. So this is going through the dichromic light and it is passing through the specimen, and then you are looking at here. So going back, excitation illumination through the specimen. So illumination is occurring through the specimen. So this is the specimen and this light is passing through, and then you are observing. Now the EP illumination. In EP illumination, this is the light source, this is the dichromatic mirror. Here excitation light goes and it will fall on the specimen here. So it is not coming from the below, it is coming from the top and then return back to your eye. So this is known as a EP illumination. Okay, so now these days EP illumination is much used in research. Why? Can anyone tell? okay, so do you have any comment on it? That why do you think that EP illumination is better in comparison to the transmitted illumination?
Some thickness for that.
This is good point. You know, you don't know the specimen is how much thick. So their transmission might be getting affected. Transmit light might be varying depending upon the thickness of the specimen. So this is good point. Okay. So however, in EP illumination there is no such restriction or not this kind of restriction is applied. So this is good point. That's why these days everything comes the EP illumination. Okay, got this point? So so one is the transmitted fluorescent microscopy, where the transmission means the light is transmitted to specimen, and in one it is falling here and then going back to your what you call ocular lens. Got this point? So this is very clear. I already told you at that day. Those who are not present at that day, they should read it again. That what are the components of the fluorescent microscope? Okay. See, this is the beautiful picture. That's the advantage. Precisely, I love this the fluorescent microscopy. I told you at that day that is the first thing which I always look for. That is a microscope, fluorescent microscope, because you can produce good picture, and that picture is more. You also feel good, and might your experiment which prove worth of your experiment. Here you can see this is the blue color is staining for the nucleus. This thread-like structure might be some actin filament or myosin filament, whatever it is, and this red structure has some another filament. So in that you can clearly visualize the internal structure of any cell. Okay, is that okay? Clear? So this is my using the fluorescent microscope means what you can achieve using fluorescent microscope. So what I achieved using fluorescent microscope, that this image is the cover page of one journal which is a bioprotocol, and this image was produced by me, where I can show different component of the cell. Then this is mitochondria, this is some cytoplasm, and this is nucleus, and then merge it. So this protocol is published on the cover page because they like this picture. So this is my picture, and also you can see here this is another picture of mine using not fluorescent microscope but confocal microscopy, where I am showing that there is some kind of protein is found nearby nucleus. So all these pictures that you are looking, this is used by fluorescent phenomena. Okay, so these which increase the impact of your finding. Okay, where you can produce a direct visualization of these cells. Here you can see this is the nucleus and this is the cytoplasm, and I am showing that in nearby nucleus there is some phenomena is occurring. So all these pictures is taken by me, and this is what published in one of the journal, that is a Journal of Clinical Investigation. Now at that day you asked one question which I told you that yes, excitation light will be always a lower wavelength and emission light is always at higher wavelength. There is a few example, Indo 1 calcium saturated, Indo 1 calcium, all these are fluorescent dye, and here you can see excitation is always low, emission is always high. Okay. Similarly, all these dyes, rhodamine green is also very, fluorescent dextran is very important, fluorescent dextran. So this is like for this dye is fluorescent dextran, TMR dextran is there. So fluorescent dextran, these are the rhodamine green, definitely green is named, so it will emit the green light. Okay, so you have to add the blue light. So this is excitation and this is emission light. So this is what these are the fluorescent dye. So some fluorescent dye name and what are their excitation and emission spectra. Okay, is that clear? Now I told you about the photo bleaching. Okay. Okay, so I already explained to you about photo bleaching. A fluorophore can repeatedly undergo the fluorescence, and it doesn't that fluorescence will always happening. It's certain after certain time it will fade. Okay, but here it is in theory it is indefinitely, but this is extremely useful because means that one fluorophore molecule can signal multiple times. The property makes fluorescence a very sensitive technique for visualization of microscope samples, even in a small amount size. In reality, this is not true, means, yeah, this is like in theory, but in reality, the fluorophore is structurally unstable due to excited light time makes susceptible to degradation. Okay, so it fluoresces every time, but when you put some light, it will just make one cycle and it will degrade. So this is known as a photo bleaching. High intensity illumination can cause fluorophore to change structure so that it can no longer fluoresce. Okay, and this is called as a photo bleaching. This is a here you can see that this is the stain cell with different fluorophore, and when you did 5-second exposure of some very high intensity light, you cannot see any color, means you can see little amount of color, but after 15 seconds of exposure you cannot see anything. Okay, it means the fluorescent dye is getting degraded. Okay, so this is known as a photo bleaching. Now, why we are come this term the photo bleaching, because our next topic is FRAP. What is FRAP? FRAP is fluorescence recovery after photo bleach. Okay, so this sounds strange that after photo bleach, how the fluorescent can recover. You got this point? So that's what we are going to understand. So I told you that photo bleaching is high intensity illumination will degrade the fluorescent molecule. So there should not be any fluorescence. But after certain time, you will see the particular area which has got photo bleach, it again start fluorescing. So then the question arises, why? Okay, so this we are going to, and that's what this technology, this technique is work on, fluorescent recovery after photo bleach, FRAP. Okay, once you do the photo bleaching, you are not expecting that fluorescence phenomena will produce again, but fluorescent phenomena again got appear. So this is called this phenomena is we are going to understand. What is this?
Plasma membrane.
This is the structure of a plasma membrane. Okay, so plasma membrane is a phospholipid bilayer. So this is lipid and this is your protein. Okay, so as an they are always stagnant and so these protein, the top one, the rounded one is a protein molecule, and this is your lipid molecule. So protein molecule will show two different type of movement. Okay, what is that? One is a lateral movement. Lateral movement is like in a lateral I am going, and another one is rotational. Rotational means if I rotate like this or swing like this. So another is rotation. So protein molecule shows two different type of movement. It is not that they are stagnant, especially in the plasma membrane. Okay, they move. Everything is really dynamic. जैसे हम इधर-उधर घूमते रहते हैं, वैसे हमारी सेल्स भी सेल्स के अंदर जो कंपोनेंट होते हैं वो भी थोड़ा-थोड़ा मूव करते रहते हैं. Especially in the cells in the plasma membrane, the proteins are moving. So these two different type of movement, lateral and rotation. So lateral we call is lateral diffusion, and if it rotates we call rotation. And this lipid bilayer will show a special type of movement which is known as a flip-flop. Flip-flop movement means it turn around and then it turn around. Okay, so just they move. So flip-flop or that they also do the transversal, means from this side to come this side. So basically it is a flip-flop movement of the lipid molecule. So protein will show only two different types, however lipid will show the flip-flop movement. Okay, so now you got this point. So the plasma membrane is the best place to understand what is the diffusion rate. So diffusion means this protein, in how much time it is diffusing and going to some another place. That's what you are going to measure, and that's what this FRAB technique is meant for. It measures the diffusion coefficient. So what is diffusion coefficient? The movement of protein molecule at particular structure you measure through the FRAB, fluorescent recovery after photo bleaching. Okay, FRAB. You got this point? Is that clear? So if you understand this one, then it is very easy what exactly what the phenomena is ongoing. Anyone in this slide, what phenomena is ongoing? एक ही बताइए बोलिए समझिए. Look it and what you can make sense of it. ऐसा हाथ उठा के पूछिए. इससे आपको मैं क्रेडिट कितने का फोटो खींचिए.
फोटो भेजिए.
आपने भी कहा देखिए दो लोगों ने कहा तो ऐसे अकेले बोलना चाहिए. This phenomenon is a high intensity light. So again let me explain. This is the red fluorescent light where you can see all these proteins are these plasma membrane proteins are appeared in red in color. Okay, and the lipid molecules are appeared in green in color. Got this point? So this is the fluorescent light where you can visualize it. Otherwise we cannot visualize it. Okay, so we stain these plasma membrane with some red and green. Got this point? Now what we do the photo bleach. We put a high intensity in this particular area. Okay, so what happens if you will do the photo bleaching? This red color will gone. Okay, so as the photo bleach, so no fluorescence is occurring. So what you are expecting? You are expecting that should not be emit any fluorescence. But what happened after certain time? Because these protein molecules are showing lateral diffusion or what are the other type of? Rotational diffusion. So they move here and there. And this particular area again showing the fluorescence. Okay. So because they are moving, and that's why this particular area they start diffusing and they reaches here and there. So then you will see again. So what happened? Recovery happened. You did photo bleaching and after certain time it got recovered. And why it is recovered? Because these protein molecule diffuses at different sites. So what happened? And the one it's come here, and so this area is again got illumination. So that's why fluorescent recovery after photo bleaching. So you got this point. So this is and there is some formula we use which to measure the diffusion coefficient of particular protein. How fast it is diffusing. So this is known as a D is a diffusion coefficient or diffusion constant. W is the radius of the beam. What is the radius? That how much the beam radius means? So this, what is the radius of this beam? Okay, radius of this beam, and then time for diffusion and how much time recovery happened. So this is known as so you can understand what is the diffusion coefficient of any protein. You got this point? Is that clear? Let's understand in another way. This is some, let's say, liver cells. So this is some cell. You did photo bleaching in a particular area. After photo bleaching, you cannot see any emitting signal. So you will see this darker. After 10 seconds or 60 seconds, it will recover again and it is showing color again. So this this is the example of photo bleaching. Now what we do, we make a graph of this one. Okay, so this graph is intensity versus time. Okay, so let's say at certain time there was a fluorescent signal was there. As soon as you will do the photo bleaching, it will the fluorescent will immediately drop down because there is no fluorescent signal is getting emitted, and after certain time it will recover. Okay, particular area will got recovered. Let's say यहाँ पे what I told you that I took out this rod and after 2 days someone changed, then recovery period of this rod is 2 days. So similar way, this slope when the fluorescent will recover, we make a graph and accordingly we will calculate. So this particular area is known as a mobile fraction. It means this much amount of this fraction of the protein is mobile. They are moving protein. Okay, and this particular area is known as immobile. The amount which is not recovered back, so that is immobile fraction. So let me show you another. So this is top view. So this is the phospholipid layer, and all the molecule is getting showing the red signal. You can see a red signal here. Now what you did the photo bleaching. So what the photo bleaching will cause? It will degrade the fluorescent molecule. So you will not see any. So it is darker in color. Here you will see a red dark patches. Now after certain time, because these are moving, so you will start this black one start displacing the red one. They are moving here and there, and after certain time you will see that this area is getting illuminated, and this is known as a frag technique. So here this is the very good. So this curve is known as a diffusion coefficient. The slope of this curve is known as a diffusion coefficient, and this area is mobile fraction, and this is the point where you do the photo bleaching, and this is the fluorescent intensity versus time. Okay, got this point? Is that clear? Okay. Now next. Now next is flip. I told you flip flop. Now don't go for the flip flop. Just fluorescent loss in photo bleaching. This is another microscopy method. You saw one frag method. Now this is fluorescent loss in photo bleaching. Fluorescent loss in photo bleaching. Now let's say what is this about? Let's say this is first the non-scientific diagram, which is this one. This one you should first understand. Okay. So what will happen? This is a cell, and this is a nucleus. So this is a large nucleus. Now what you did, you bleached particular area by putting some high intensity light and measuring on this area. So this is different. उस खिड़की से कुछ कर रहे हैं, देख रहे हैं, ऑब्जर्व कर रहे हैं इस एरिया को. So and you continuously bleach this area until unless this area is getting, they will lose all the fluorescent. Okay, so continuous photo bleaching so that this area is also do not emit any fluorescent signal. Okay, so this is known as a fluorescence loss in photo bleaching. So what exactly happened? See, this is the bleached. This is your plasma membrane, and this area is a bleached area, and this is the measurement area. Okay, so what you are continuously emitting the light. So even though, so you are bleaching this particular plasma, but they will show the diffusion. They will go there. Then the new molecule will come and it will again get fluorescence. So it means the entire movement, as soon as all the protein will come at this place and they will get photo bleach, so that you will not see any kind of signal. Is it understandable or not?
Yes, sir.
So it will take time. It will take time. So the entire protein will come in a particular area which is getting photo bleached. And there is a one time where entire area will get photo bleached. You will not see any kind of signal. So this is known as a fluorescence loss in photo bleaching. So this protein molecules are bleached as long as they are mobile and not permanently encode to the cytoskeleton or extracellular matrix. So this is another this is flip method where you are looking the total diffusion of all the protein, how much all the protein is getting diffused in a particular area. You got this point? Does it make sense? आप समझ गए ना? Means at least you understand the how this microscopy works. You are continuously emission until unless you will the photo bleached area is not the measured area. The measured area is different and photo bleached. Like let's say if okay now. So, what is the difference between trap and flip? You already understand that fluorescent molecule bleached in a small area using laser beam. The fluorescent intensity recover as the bleached molecule diffuse away and unbleached molecule diffuse into the irradiated area. The diffusion coefficient calculated by a graph or rate of recovery. The greater diffusion coefficient is the faster method. So, this is about your trap. Now, flip. Small area of membrane is irradiated continuously and fluorescent membrane in a separate area. Fluorescence in the second area progressively decreases as fluorescent protein diffuses in. Eventually, all fluorescent protein molecule bleach as long as they are mobile or not permanently achieved to the cytosol or extracellular matrix. As long as they are moving, you will see the entire protein molecule will get bleached. Got this point? This is the difference between flip and trap. Is it okay? Now third is this is different type of all our fluorescent. The microscopy is all this microscopic method or microscopy method is based on the emission of the fluorescent. Now the third one is FRET. So let's say what is a FRET? So in FRET is the full form of FRET is fluorescence or Foster resonance energy transfer. Okay, so what is the full form of the FRET is fluorescence or Foster resonance energy transfer. Okay, so if you understand resonance energy transfer, okay, resonance energy transfer of resonance energy will cause the fluorescence. This, if you want to understand this FRET phenomena, you should understand in this way. Resonance energy transfer is causing fluorescent phenomena. Okay, so here there is a two molecule. This is one protein molecule and this is another protein molecule. So what exactly we are doing here? If generally this protein molecule, when they are apart less than 10 nanometer, only then you will see a FRET phenomenon. If they are more than away, means their distance between these protein molecules is more than 10 nanometer, it will not show the FRET phenomenon. Got this point? So let's understand. Similarly, how two magnets will only attract each other when they are close enough. You know that एक magnet, we kept one magnet at one end, another, so they will, only when they are close, then they will show the magnetic property, otherwise they will not. Fluorescent energy can also be transferred between fluorescent molecules if they are extremely close proximity. This principle is behind the fluorescent or Foster resonance energy transfer, which is a technique used to determine whether two fluorescent molecules are within certain distance of one another. So definitely they are tagged with some protein molecule. So once this you will see this phenomena, then you will understand that, let's say I tag protein A with some dye, I tag protein B with certain dye, and then you will see that protein B is emitting the fluorescent signal. Then you will understand that protein A and B are in a close proximity. Okay, so this is in this phenomena you will see the protein-protein interaction. Now proposed by Theodor Foster. So why is we call Foster resonance? Because of the scientist who discovered this phenomena. So proposed by Theodor Foster in 1946 and confirmed experimentally by Stryer and Hogland in 1967. Fred takes advantage the principle of energy transfer between donor and acceptor fluorophore. So donor fluorophore and acceptor fluorophore. So one is known as a donor fluorophore, and those who accept it is known as an acceptor fluorophore. Molecule which is occurs once are within a certain distance of each other. So if we want to understand the Jablonski diagram, so here you can see it is a donor fluorophore where when it gets excited, certain energy fall here, so it will go into the higher excitation state, and when it gets down it will emit some signal, and then this signal will taken by this acceptor fluorophore, and if at certain proximity, if it is close, then this emission will be taken by this one and it will excite again, and it will emit the another fluorescent, it will show the another fluorescent, or it will emit the another wavelength of the light. Okay, got this point? Now the donor fluorophore absorbs proton and jumps the ground state. So this is the exactly whatever I am showing here, you can make it. So there is a very two popular dye which has been used for this Fred experiment. One is CFP, another is Venus. Okay, so CFP is known as a cyan fluorescent protein. Okay, cyan fluorescent protein, and another one is yellow fluorescent protein. Okay, so this will emit the cyan color, cyan fluorescent protein, and this is YFP protein. It is also it is known as a Venus. So when they are apart, 10 nanometer apart, this fluorescent molecule will not fluoresce. But when they are in the close proximity, it will fluoresce. Okay. So in this way, we can see the protein-protein interaction, and also so here we can see when they are distance apart, this donor emission will not overlap into the acceptor in excitation, so thereby no Fred is there. But when the donor emission overlap between this acceptor excitation, it will show the color. Okay, so is that okay? You got this point? So correct orientation. So this again, the orientation should also important. If they are close, but they should be oriented in a proper way. So I hope it's clear to all of you. It is basically for protein-protein interaction. Right. So again, this is showing that this is excitation. CFP and YFP protein, you tag the one with the cyan fluorescent protein and yellow fluorescent protein with the protein AB, and if they are in a close proximity, this will emit yellow color. Okay. Okay, so now there is another method which we are going to read again, that is the fluorescent correlation spectroscopy. Okay, so we already covered the fluorescent microscopy, we already covered the frap, then we covered the flip, and then we covered the fret, and then last is left is fluorescent correlation spectroscopy, which we will cover tomorrow. Okay, so and this method is based on the fluctuation, that how a fluorescent molecule is fluctuating into a particular area. So that we are going to see tomorrow. Otherwise, you will might be overloaded with different type of